Oh, I'm Connie, and I'm going to be talking today about using genomic barcodes for discriminating complex species. And to give a little bit of background, traditionally, to use DNA to identify plant species or animal species, we look at really little bits of DNA that don't change very much in evolutionary time, so that members of the same species all have the same sequence of DNA bases in these short little sections, and those are called DNA barcodes. And there are lots of different ones that you can use. For animals, there's pretty much one that works on everything. And for plants, that's not so much the case, but we have several that we use. However, there are still quite a few plant species that don't really work with these DNA barcodes. They either all have the same, like members of lots of different species all have the same DNA sequence in DNA barcodes, or members of the same species all have very different sequences in the DNA barcoding loci. So for this project, I'm taking two target groups to investigate designing new genomic barcodes, so new sections of DNA that we could use as barcodes to identify groups to the species level. And then from this, we're hoping to better understand the genetic nature of species differences. And to do this is a pretty big question, the genetic nature of species differences. And we're kind of, to use Pete's words, taking a sledgehammer to crack a nut. And we're capitalizing on the decreasing costs of whole genome sequencing to design data efficient and cost efficient ways of telling species apart. Yeah, so to do that, we've chosen two groups, which are potamogetans, the pondweeds, and salix, willows, which I'll talk about in a minute. So for potamogetans, traditional barcoding loci can't tell all the species apart. They're a morphologically pretty diverse group, especially the narrow-leaved pondweeds tend to be more complicated to identify, especially when it comes to identify morphologically, especially when it comes to the question of hybrids. They are an important freshwater ecosystem and they're bioindicators of freshwater systems. And there's been growing interest in using eDNA, so that's sequencing the DNA that's found in water bodies to identify the species present. And it would be useful to be able to identify the potamogetan species, the pondweed species in an area using eDNA. And currently, seeing as the barcoding loci don't really work for it, the eDNA assays aren't like totally effective, although it's been shown that some of them do work for some species. So yeah, the two groups that I've selected are potamogetan and salix, as I said. Both groups have multiple closely related species. They are important for biomonitoring and biodiversity characterization, and they both have manageable genome sizes. And to put into context what I mean by a manageable genome size, I'm here talking to you with about 30 million base pairs of DNA. And the mistletoe, which has an absolutely massive genome, has 90 billion base pairs of DNA. So I'm definitely not dealing with anything like that. And Salix and potamogetan have approximately 500 million to 1.5 billion base pairs, so much more reasonable. And for the salix, we're looking at the montane, Arctic, and subarctic salix, or some species of it. Traditional barcoding loci often fail for salix. They are an ecologically and economically important ecosystem, and the montane scrub willow is very important here in Scotland, but also a highly threatened system. And they're important paleoclimate reconstruction species. So when they take the ancient ice cores in the Arctic, for example, you often get a lot of like willow seeds or evidence of willow there. And although you can identify the species from the seeds, it's pretty difficult. And it would be great to just be able to use like an easy DNA assay to tell you what species of willows were there. And I think this paper is just an absolutely brilliant way to show the problem in willows. As you can tell from the title, the spectacular failure of DNA barcoding in willows. And although the figure looks a bit complicated, basically what it's saying is you've got tons of different species of willow there, and you cannot tell them apart just by looking at the traditional DNA barcoding loci. So how am I going to address this problem? So I am taking 20 species from each genus. So that's all the UK species of Potamogetans and then a subset of the willow species. And then I'm aiming for 5 samples from each species from geographically distinct populations. So they're going to be from very different populations around mostly the UK, but then we're also getting some species from throughout Europe. And then my pipeline is we're collecting samples, I'm extracting the DNA, we're sequencing the whole genome first, and then we're going to do some analysis to find informative regions within the DNA that hopefully we can use to identify to the species level. And basically we're trying to work from the most amount of data possible, the whole genome, and then we're moving down to the smallest amount of DNA that will tell us the most amount possible. And then I'm going to try and turn that into an assay, like a little test we can use in the lab or from stuff collected from the field that will beautifully tell us all the species apart perfectly with no issues. And where I am to date is we've collected all of the UK species of pondweeds. For some of them, we've only got 3 populations because there are some very rare pondweed species like Potamogeton epihydrus that actually only has 3 populations in the UK. But for all the others, we've got the 5 geographically distinct populations. Out of the 20 willow ones we were aiming for, we've got 12 completely fully sampled. So we've got actually for those we've got well over 5 geographically distinct populations. The others we're some we've got nearly there, like 4 or 3, and then for a couple we're still waiting for the samples from some collaborators. And I have a little bit of preliminary data to show you here. With 47 samples have already been sequenced. We've already sequenced 47 samples of 6 species of willow and 10 samples from 8 species of pondweeds. And the type of sequencing we've used is at the bottom there. If it means anything to you, great. If it doesn't, just ignore it. And here is the full genome of the Salix species, the willow species. Basically, the colours are the species, and they're all very prettily grouped together. So the same species are all clustering with their friends really nicely when we look at the whole genome. When we look at just the plastome, which is like a little bit of DNA that we often use to identify to species level, you can see that all of the colours are not nicely with their friends. It is not a good tree there, phylogeny. So that's not great for the willows. And then here, ITS, which is another region that we often use to identify species. It's a bit better than the one before, but you can still see, especially with Salix arbuscula, Salix liponum, and Salix herbacea at the top, pretty messy. So this is also not good. So then we've added some more DNA. And this is a group of DNA regions called the Angiosperm 353 bait set, which, as the name possibly suggests, it's got 353 little bits of DNA regions. And I have pulled those out of the whole genome, and it does fairly nicely identify to the species. You can see that all the colors are clustered together again. It's not perfect, but it's better. So this is a point that we could start trying to remove some of those loci and work down and see how much we can take out before it stops working. So the next steps, as I said, take out data, downsample, repeat what I have just showed you for the Salix for the Potamogetans. That's in process at the moment. I really wanted to be able to have some figures to show you on Potamogetans, but it didn't quite come together in the end in time. Finish extracting the DNA from the samples we've collected, sequence that DNA, and then start looking for informative regions of DNA in all the species that we can turn into an assay. For these, we've also got for both groups, we've got the issue of hybrids and polyploid genomes, which are big genomes with multiple chromosomes. chromosome pairs. And then, so it's sometimes difficult to assemble the genomes for that. And for hybrids, we want the assay that we design to also be able to identify hybrids. So yeah, we will be testing the assay out on hybrids and then adapt the assay to work for eDNA for potamogetans. And just in a little bit on eDNA, here's a paper showing that eDNA can effectively identify some potamogetan species already. And it's so cool that you can just basically take like a cup of water and then identify the species that were in that water body from the water, just looking at the DNA that's in it. So hopefully we'll be able to come up with a good assay to add to this. And lots of BSPI members have helped us with the collections for these groups. And I'm hugely grateful to everyone, anyone who has sent me samples. I've been annoying lots of people, giving them little bags of silica gel, being like, please, if you see a pondweed or a salix, please get some samples. So thank you very much to everyone who has helped. Happy to answer any questions.
Any questions for Connie? One at the back, just there. Sorry, at the back there, we're going to try and choose people that are the furthest distance apart from where you're standing for the questions. So we need one from the back row up there in a second.
I wasn't expecting to talk about potamogetan and genetics. That's really exciting. What a great project. I wanted to ask about the loci. What is it that's not working with the traditional barcoding regions that used? Is it kind of primer annealing, or are they too conserved or not conserved, the regions?
In the potamogetans?
Yeah, and in the salix too.
In the salix, they tend to all have identical barcoding regions. There's not enough diversity. In the potamogetans, everything will kind of work up to a point, and then there's always something that's weird and clusters with something else. So there's, again, not a huge amount of diversity within the traditional barcoding loci, and there's nothing so far that's been tested that works routinely for all the potamogeten species.
Okay, brilliant. Thank you.
Thank you very much. That was fascinating. How long do you think it'll be before we can send you a hybrid potamogetan and you can tell us what it is?
Oh, please send me a hybrid potamogetans. I'm going to need some to test on next year. Well, my contract finishes in 2026, so definitely by then I should have something. I'm hoping to be testing an assay out in the hybrids next year sometime. So yeah, I will gratefully accept hybrids to look at.
Thank you. About how long does it take to sequence an entire genome, let's say, from the salix? Do you have an idea?
You mean from me collecting the sample, extracting the DNA? I mean, the limiting factor is me, but not very long. I mean, I can extract the DNA in a day, and we send it down to Cambridge where it's sequenced, and they'll have the data back to us within 2 weeks to a month.
All right.
And then I have to assemble, because it's short read sequencing, so it's like a little jigsaw pieces, and then I have to put the jigsaw pieces back together in the right order, which can be a little slow in the species that don't have a reference genome. But yeah, about a month, I guess, to get the data back.
We don't have facilities here in Scotland for that, correct?
Oh no, we do. There are definitely sequencing companies you can send to here in Scotland.
It's perhaps worth making the distinction between sending material off to have the whole genome sequence, which is a kind of complicated and in-depth bit, and the assay, which is just the final endpoint, which is a much faster turnaround for the identification bit.
Yeah, for the and like the outpoint of an assay, you would just get the DNA, run the assay, and then the results would be able to very quickly tell you what species it was, ideally. No complicated jigsaw piece putting back together like I'm doing at the moment.
Doug, if you can use the microphone just so everybody else can hear the question.
I take it dried herbarium specimens are of no use in the study you're doing.
So we haven't used any herbarium specimens for the samples so far because we want really high quality DNA to look at the whole genome. It might be possible that the assay would work on herbarium specimens. We'd have to test that. I guess if I had time, that's something we could look at. But in terms of samples to like test the assay on, for example, silica dried material would be best.
Thank you.
So thanks, Douglas, for asking the question I was always going to ask there. That's superb. And I'm very conscious the head of the herbarium sitting right behind me. But do you think, is there any merit, given there's quite a lot of hybridization going on there, with the samples you've got, it looks like there's hints of sort of hybridization in amongst all the populations, maybe? I don't know if it's easy to tell, but is it worthwhile looking at the assay on the type specimens just to verify that it behaves?
Yeah,
as well
I
as
mean, we
possible?
absolutely could do that if we were allowed to look at type specimens. Yeah.
Can I?
Yeah, please.
I mean, I think we tended to stay away from the type specimens over past years because the levels of failure were so high. But it was relating to Douglas's question, with the new methods, the levels of success with herbarium specimens is so much better. Olwyn, as the curator of the herbarium, is very receptive to type specimens being sequenced where there's high confidence in the assay. And indeed, there's a program at the minute, which is a sort of collaboration with Kew and the Natural History Museum on sequencing fungal types that's just got underway very recently. So that's a big shift. And a few years ago, it had been no, because you'll take something from the specimen but not add something in terms of data. Now that position has changed.
With the dioecious species like Salix, can you tell the difference between males and females of the same species?
I think
Hadn't you thought of it?
people can, and from the DNA I could. If you handed me a Salix and asked me if I knew from looking at it morphologically, the answer would be no. Whether the assay would be able to tell that, I don't know. It depends on the DNA regions that it ended up targeting. It's probably unlikely.